Of a TCM-derived agent aimed at rejuvenating aging hematopoietic stem cells with predilection for lymphoid differentiation. Thank you, Dan.
Thank you, Chairman, for your introduction. Good afternoon, ladies and gentlemen. My name is Dan Gao. I'm from Xuanwu Hospital, Beijing, in China. It's my honor to be here for this presentation. Today, my topic is identification of a traditional Chinese medicine-derived agent aimed at rejuvenating aging hematopoietic stem cell with predilection for lymphoid differentiation. As we all know, the problem of aging population becomes more serious. According to the nation's projection, we can see that by 2059, the proportion of 16 will reach about 25%, and the future will continue to be aging. So it's very important to improve aging and related disease. As reported, aging clock is regulated by many factors such as stem cell exhaustion, mitochondrial dysfunction, genomic instability, and telomere short, and so on. However, many experts believe that stem cell aging is the root of human aging. Actually, hematopoietic, here we called HSCs for short, occupied an extremely important place in stem cell, which has ability of cell renewal, proliferation, and differentiation into myeloid and lymphoid progenitor, leading to the balanced production of both myeloid and lymphoid lineage to maintain the homeostasis of blood lineage and the immune system of our body. However, with aging, HSCs result in abnormal self-renewal and differentiation, like decline in regeneration capacity, decrease in the lymphoid lineage, and increase in the myeloid lineage. Therefore, it's so significant to explore an intervention that can effectively improve HSC aging, especially lymphoid failure. Traditional Chinese medicine has a long history of anti-aging. As we see, Bianqi, a famous doctor of Chinese medicine in ancient time, is said to live more than 300 years, and many Chinese herbs were recorded have anti-aging effects. So in this study, we first sequenced 9 active compounds from different herbs of anti-aging according to the clinical trials, reference, and ancient books in total body irradiated mice based on the CLPs and B cells, which was acute mouse model possessed aging HSCs phenotypes similar to naturally aging mice. We found that only one compound, TSG, could increase the number of CLPs and downstream B cells. TSG is one kind of steel-binding glucoside from Xuanwu, which has a long and profound history in prolonging life. Then we performed a comprehensive assessment on potency of HSC's rejuvenation in naturally aging mice. This is Xuanwu. In this study, 20 mice were treated with vehicle or TSG by gavage daily, which were analyzed after 30 days by FACS. It was found that mice that were treated with TSG present a significantly higher number of CLPs in the bone marrow per mouse, and also great higher number of total B, pro-B, and pre-B of the B cells lineage compared to aging mass, but didn't produce any substantial effect on the number of T and myeloid cells of the bone marrow. Nevertheless, TS treatment results in significant elevation in the total number of 3 subset of T cells or cellmers and tend to restore balance as a large increase in the percentage of T cells and percentage of B cells and a decrease in the percentage of myeloid cells, but had no effect on the T cells in the peripheral blood. Moreover, TSG had no effect on the number of HSCs, including long-term HSCs and short-term HSCs, and common myeloid progenitors. We also detected ROS apoptosis proliferation in the LT or CLPs to investigate the potential cause of lymphoid increase after TSG treatment. We found that the percentage of BRDO positive CLPs, NAC5 positive CLPs that were unaltered in comparison to the aging mass treated with vehicle. The level of ROS was the same situation after TSG treatment. As we know, aging HSCs were also known to exhibit greatly reduced repopulation potential. Furthermore, we sorted HSCs or CLPs from different groups of mass and transplanted to the receptor mass to explore the influence to repopulation potential after TSG treatment. We found that the mice that received the long-term HSCs from the TSG-treated aging mass portrayed the hair contribution in all the cases, and the same phenomenon was observed in the CLP's transplantation. To explore the mechanism of TSG for anti-aging, we applied transcriptal analysis. It was found that the transcriptome of HSCs from the TSG-treated aging mice exhibited a profile analogous to that of the young mice. Gene set arrangement analysis and heat map analysis of 36 genes strongly tied to lymphoid lineage revealed that lymph-associated genes were significantly enriched in the TSG-treated aging mice. Some key genes were verified by RT-qPCR. Moreover, in the transcriptional analysis, some cell metabolism genes, such as oxphosphorelated genes and HSC's quiescent genes, were regulated by TSG. We can see that oxphosphor gene were down-regulated and the quiescent gene were up-regulated after TSG treatment in the LT of mice. Then we used seahorse to examine the metabolism of HSCs in the bone marrow. We can see that TSG could induce the production of ATP and keep the HSC state in the GO phase. Then we guessed MPK as the energy sensor, if it could be influenced by TSG to play the role. So we examined the protein expression by Western blot in HSCs. We found that the protein level of activated AMPK and its downstream of phosphorylate ACC were increased significantly, so TSG might maintain aging HSCs quiescence and activated AMPK. Then we treated mice with MPK inhibitor compound C by TL1 injection every other day. The results showed that compound C blocked the protective of TSG on the number of cell pieces and its downstream B cells in the bone marrow, as well as the proportion of B cells in the peripheral blood. For HSC transplantation, as shown in D and E, the percentage of donor-derived HSCs in the bone marrow of TS-treated aging mice were also damaged with the treatment of compound C. Overall, the results suggest that TSG can effectively enhance HSC function and lymphoid differentiation, mainly through the activation of AMPK. Further, we also applied in vitro analysis to verify the mechanism of TSG for HSC aging via activating AMPK, such as molecular docking, SPR, and promega enzyme activity assay. We found that TSG could bind well to AMPK. Maybe AMPK alpha 1 is a little better than AMPK alpha 2. The kinase assay was performed to measure luminescence-based AMPK activity. Clear activation of AMPK were triggered by TSG with dose-dependent manner. More importantly, we studied the effect of the TSG AMPK activity using specific AMPK inhibitor compound C and AMPK agonist ECA on synthetic cell lines. Using the Washington blot, we found that AMPK and ACC phosphorylation levels were ultimately increased in a dose-dependent manner after TSG treatment, which were even a little better than the ECA. Combined compound C with TSG, phosphorylation of MPK and ACC were dramatically decreased compared to TSG treatment. Taken together, these results indicate that TSG might promote MPK activation directly. Previous studies have shown that MPK activation is crucial to maintain the type 2 stability, and the type 2 as the one kind of demethylation enzyme was a substrate of MPK. Here we found that TSG treatment effectively boosted the protein level of active MPK and type 2, which were attenuated by the coordinated administration of AMPK inhibitor compound C and TSG in the aging mice. We also found that in cell culture above mentioned. Furthermore, we evaluate genomic-wide DNA methylation of long-term stem cell isolated from the bone marrow of mice. Cluster analysis of the methylation profile revealed that the LT from the 4 groups fell into 2 categories: young and TSG groups clustered together, and old, as well as old and old TSG combined compound C group clarified into the same cluster. Specifically, the methylation level of lymphoid regulated gene of the LT showed an evidence decrease in TSG-treated aging mice, but were found elevated in TSG and compound C co-treated aging mice. To further validate this point, we adopted type 2 mutant mice and found that TSG treatment failed to increase the absolute number of cell pieces in total body irradiated type 2 mutant mice. We next verify the role of TAT2 by co-culture assay in vitro. The results show that HSCs sorted from the bone marrow of mice exhibit enhanced differentiation potential to lymphoid cells after TSG treatment, while TAT2 inhibitor Bobcat 339 significantly decreased lymphoid differentiation potential when co-culture with the open stromal cells. All above implies that TAT2 as a substrate of MPK has an imperative role in driving HSC differentiation towards lymphoid lineage. Collectively, in this study, we try to devise a new system for assessing a potential of individual agents for rejuvenating agents, HSCs. We successfully identified the compound TSG that exhibit potent efficacy in rejuvenating aging HSCs in aging mice and total body irradiate induced premature senescence. We deemed the MPK TAT2 acts as a potential mechanism of facilitating describing the metabolism to epigenetic signaling pathway for rejuvenating aging HSCs. That's all. Thank you. Thank you to all the audience, to my tutors and my teammates. Thank you.
Thank you, Dr. Gao. The paper is open from just one question in view of the time. If there is no question, then we move to the next speaker. Thank you, Dr. Gao, again.
Thank you.
Is Dr. Ilari Mäki-Opas in the room?
Yes.
We welcome you, Dr. Mäki-Opas. He will present us the paper TRPA1 as a potential factor and drug target in scleroderma, dermal fibrosis, and alternative macrophages activation are attenuated in TRPA1 deficient mice in experimentally induced scleroderma.
Thank you for the introduction, and good afternoon, everyone. I'm very humbled to have this opportunity to talk here in this great congress today. And my name is Ilari Mäki-Opas. I'm a physician from Tampere, Finland, and I'm doing my PhD thesis in the immunopharmacology research group led by Professor Eeva Moilanen in Tampere University. So my talk's title is TRPA1 as a potential factor and drug target in scleroderma, dermal fibrosis, and alternative macrophage activation are attenuated in TRPA1 deficient mice in experimentally induced scleroderma. So quite a long one. And there, under the title, are the names of other writers of this work. First, a brief background of the three main subjects that this work crosses over. First, on the left, scleroderma. It is a fibrotic skin manifestation of systemic sclerosis, which is a fibrosing rheumatoid disease affecting 1 to 4 people in 10,000. There in the middle, macrophages. Macrophages are known to have this spectrum of different phenotypes, two opposite ends of which are the classical activation, abbreviated M1 most commonly, and it's the pro-inflammatory phenotype of macrophages. The other end are alternatively activated macrophages, M2 macrophages that are most famously anti-inflammatory, but they are also markable in resolution and wound healing, and their kind of overactivation might lead to unwanted effects like fibrosis. And it is known that in pathogenesis of scleroderma, M2 macrophage activation is included. Then, on the right, the TRPA1 receptor transient receptor potential anchorin 1. It's a non-selective cation channel, activation of which causes infuse of calcium ions, and that causes pain and release of inflammatory mediators. It is originally known to be expressed in neurons, but now more and more papers showing its expression in other non-neuronal cells as well as like skin cells and inflammatory cells. So we now know it's not only neuronal channel. So how do we wrap up these three subjects together to make a hypothesis? Earlier studies have shown that TRPA1 deficiency has an impact on models of M1-driven diseases like atherosclerosis and hypoxia reperfusion injury. Then, as I said earlier, systemic sclerosis is an M2-driven disease, as shown in this figure on the right-hand side. It is also interesting that TRPA1 has been shown to mediate Th2-driven inflammation, which is closely related to the M2 macrophages. So, we hypothesized that TRPA1 deficient mice should have attenuated manifestations in experimental models of scleroderma. So, we first sent out to study the role of TRPA1 in experimental model of scleroderma in vivo, and for that we used bleomycin-induced scleroderma in wild-type and TRPA1 deficient mice. Then we wanted to investigate the underlying M2 macrophage-mediated mechanism in vitro, and there we used J774 mouse macrophages, cultured and commercially available. And then we also harvested mouse peritoneal macrophages from the wild-type and TRPA1 deficient mice from our own lab. And now on to the results. From now on, you can see on the upper left corner, there's a box indicating what the current research subject was. So we found that the TRPA1 deficient mice have attenuated bleomycin-induced dermal thickening. So on the left here, we can see the demonstrative histology cuts, HE colored cuts of mouse skin that is bleomycin injected on the bottom side of wild-type and TRPA1 deficient mice, and here are the control counterparts of both genotypes. As here, we can see the bars presenting the mean skin thickness. We can see that in the bleomycin-treated mice, the TRPA1 deficient mice skin didn't get thicker as much, so there's some form of downregulation there. Second finding was that bleomycin-induced collagen accumulation is attenuated in TRPA1 deficient mice. Here again, demonstrative histology cuts, but now colored differently with Masson's trichrome staining that highlights collagen. And here, these bars show means that indicates the collagen density in the skin, and we can see that the TRPA1 deficient mice have skin less dense in collagen compared to the wild-type animals. We also took the skin samples and extracted messenger RNA from them and ran RT-qPCR to see expression of 3 of the most common collagen types in skin, and we can see that also in mRNA level, the expression of collagen is downregulated in TRPA1 deficient animals. Here, we show that bleomycin-induced expression of profibrotic mediators is also lowered in TRPA1 deficient mice. So these are same skin samples as previously with the collagen pictures. There are many profibrotic markers here, and then also a couple of more classic inflammatory mediators like interleukin-6 and cyclooxygenase 2. The profibrotic factors on the upper row, as you can see, show that the TRPA1 deficient animals had lower expression of these profibrotic factors. Then, next finding, markers of M2 type macrophage activation are lower in bleomycin-treated TRPA1 deficient mice. So here we can see these are the 2 two main markers of M2 macrophage activation. In mice, I have to say, it's a bit different in humans, but in mice, these two are the main ones. And then interleukin-13, which of course belongs to Th2 lymphocyte or like inflammation family. So we can see that these M2 type macrophage markers were substantially downregulated in TRPA1 deficient animals, and also was the Th2 related interleukin-13. Now on to the in vitro results. Cultured J774 mouse macrophages were incubated in bleomycin in absence and presence of interleukin-4, which is a main cytokine of Th2-mediated immunity and a main inducer of M2 macrophage polarity. For the first time, we saw that bleomycin by itself dose-dependently increased the expression of M2 markers, arginase 1 and MRC1, as seen in these white bars over here. And when interleukin-4 was present, bleomycin greatly enhanced the effects of IL-4, suggesting an interaction, as seen in these gray bars over here. In this picture, on the left, there is a sum-up figure of intracellular mechanisms of macrophage polarization. M2 mechanisms are on the green side on the bottom here, and two of the main intracellular mechanisms of M2 polarization are phosphorylation of STAT6 protein and expression of PPAR gamma. From the J774 cultured mouse macrophages, we studied these two things, and here we can see that in case of STAT6, there is no difference. The bleomycin makes no difference here, but to the PPAR gamma expression measured as mRNA, there is the bleomycin somewhat boosts the effect of interleukin-4, which is best seen at 4-hour time point. To see if TRPA1 got anything to do with the M2 promoting effects of bleomycin, we harvested macrophages from the peritoneum of wild-type and TRPA1 deficient mice, cultured them and incubated with bleomycin and interleukin-4. We saw that interesting responses to IL-4 alone were significantly downregulated in TRPA1 deficient mice regards to M2 markers arginase 1 and MRC1, and also Th2 interleukin-13. With the M2 markers, we can also see that the marker expression boosting effect of bleomycin is somehow TRPA1 mediated, as you can see by comparing these two black bars and these two black bars over here. So finally, to conclude, these results show that TRPA1 deficiency downregulates bleomycin-induced processes like skin fibrosis, collagen accumulation, M2 macrophage activation. Bleomycin has intrinsic effects on macrophage polarization that are TRPA1 dependent, and therefore we think that TRPA1 antagonists could potentially be future drugs used in M2 macrophage-driven diseases like systemic sclerosis. And finally, I want to thank all the funders as well as all my colleagues at the Immunopharmacology Research Group. If you're interested, the article containing this data was published between the abstract submission and this Congress, so if you want to take a look at this open access article, there's the QR code you can scan. Thank you very much for your attention.
Thank you very much, Dr. Mäki-Opas, for your presentation. The paper is open for questions. I have one. In your model of scleroderma, have you characterized any agonist of the TRPA1 receptor that could justify the involvement of this receptor and the response to bleomycin?
So do you mean if we use any TRPA1 agonists in the model?
No, if there is, when you induce scleroderma in your mice by bleomycin, if you have the increase of some endogenous agonist of this receptor that could explain why the knockout mice for these receptors have a better response.
Yeah, we haven't done any analysis like that to like any oxygen or nitrogen species or any other endogenous agonists. We haven't done it, but that's a good idea and for the future. Yes.
Okay. Well, if there is no further questions, we thank you very much for your presentation. And I'll call now the next speaker is Dr. Elisa Porchietto from the University of Camerino. Is she in the room?
I'm here.
Oh, sorry. And she will present the paper Tetrabromobenzotriazole counteracts metabolic derangements evoked by the consumption of hypercaloric diet in mice.
Good afternoon, everyone. I thank the organizing committee for the opportunity to present here my activity research. During the last year, we have witnessed a lifestyle modification characterized by the increased consumption of unhealthy food and reduced physical activity that have spread the insurgence of metabolic disorders such as obesity and type 2 diabetes. This pathological condition share the common feature of a low-grade chronic inflammation, also known as metaflammation, that is becoming a new interesting field of research for counteracting metabolic disorders. Growing body of evidence suggests the role of kinases as crucial mediator for the activation of inflammatory cascades. As we could see from this graph, the correlation between kinase and obesity and kinase and type 2 diabetes has been increasing over the last 40 years, especially during the last decades. In the light of this, several small molecule kinase inhibitors were developed, and nowadays around 90 small molecules have been approved by FDA. The laboratory of Professor Massimo Collino from the University of Turin has already been proved how targeting inflammatory cascades is effective in ameliorating meta-inflammation. In these two works, they demonstrate how the high-fat diet consumption evoked the overexpression and the overactivation of NLRP3 inflammasome complex and JAK2 kinase, and how the pharmacological inhibition of these two targets is effective in reducing glucose intolerance and the higher level of pro-inflammatory cytokine in the serum of high-fat diet fed mice. In the light of this, it is well known how these inflammatory cascades are specifically activated by post-transcriptional modifications such as phosphorylation, which are mediated by kinases. Among them, we identify a crucial serine-threonine kinase named casein kinase 2, whose pathological role has already been proved in several pathological contexts. In particular, casein kinase 2 is directly involved in the activation of two well-known pro-inflammatory cascades, NF-κB and JAK-STAT, involved in meta-inflammation. For this reason, we selected a potent CK2 inhibitor named 4567 Tetrabromobenzotriazole, or TBB, and assesses effectiveness in a murine model of high-fat diet consumption. In detail, mice were randomly divided in 3 groups. One group was fed with a controlled diet for 12 weeks, while the other one with an high-fat, high-sugar diet. A subgroup of the high-fat, high-sugar fed mice received TBB at the dose of 2.5 milligram per kilo for the last 8 weeks of the protocol. Despite not being effective in reducing body weight gain evoked by high-fat diet consumption, TBB improved diet-induced glucose intolerance, as we could see from this data, representing a reduced fasting blood glucose level compared to the high-fat, high-sugar fed mice, and a lower area under the curve following oral glucose tolerance test. Moreover, TBB administration ameliorates lipid plasmatic profile. As we could see, both triglyceride and total cholesterol level were dramatically reduced compared to the high-fat, high-sugar fed mice. Next, we evaluated some biological marker of organ damage, such as the transaminase. Both AST and HALT level were massively increased in the high-fat, high-sugar fed mice, and TBB present lower levels in the plasma of the TBB-treated mice. Next, we want to evaluate the TBB effect in alleviating local inflammation at hepatic level. First of all, we evaluated the expression of a well-known adhesion molecule, ICAM1, which is known for recruiting immune cell in the inflamed tissues. As we could see, ICAM1 was overexpressed in the high-fat, high-sugar-fed mice, while TBB-treated mice presented lower expression of this molecule. Next, we want to characterize the immune subpopulation that was recruited in the hepatic tissue, and by the evaluation of myeloperoxidase, which is an enzyme involved in the neutrophilic infiltration. Both the expression and the activity of this enzyme was statistically significant reduced in TBB-treated mice compared to the high-fat, high-sugar group. Subsequently, we evaluated the drug target, CK2, in the hepatic tissue. As we could see, the high-fat, high-sugar diet evoked a massive increase of the catalytic subunit alpha, which is responsible of the phosphorylation of the target, compared to the treated mice. Next, we evaluated the role of the kinase in the crosstalk with the well-known pro-inflammatory signaling cascades such as JAK-STAT and NF-κB, and the potential beneficial role of TBB administration. First of all, we evaluated how TBB could affect the NF-κB signaling pathway. As we could see from this densitometrical analysis, the high-fat, high-sugar diet evoked an overactivation of both IKK and IKB kinase, along with an increased nuclear translocation of P65 subunit. TBB group, the TBB-treated mice presented a statistically significant reduced overactivation of both IKK, IKB, and a lower nuclear retention of P65 subunit. This data was further confirmed by the mRNA expression of TNF-alpha that was dramatically increased in the high-fat, high-sugar-fed mice compared to the treated one. Next, knowing the direct role of NF-κB in promoting the genetic expression of NLRP3, we evaluate whether the pharmacological inhibition of CK2 could be effective in reducing the overactivation and the overexpression of NLRP3. As we could see from this data, both NLRP3 and cleaved caspase dramatically increased by the high-fat diet consumption were statistically significantly reduced in the TBB-treated mice. In parallel, we evaluated the interleukin-1 beta mRNA expression, and as we could see, the TBB treatment was effective in reducing the diet-induced increase of this pro-inflammatory cytokine. In conclusion, we evaluated the effect of TBB administration on JAK-STAT pathway. We confirmed that this pathway is overactivated by the hypercaloric diet consumption, and that the CK2 inhibition was effective in reducing the overactivation of these pro-inflammatory cascades. This data was further strengthened by the evaluation of mRNA expression of interleukin-6, which is known for promoting the activation of these signaling cascades. And again, the TBB treatment was effective in reducing interleukin-6 mRNA expression. the reduction of local inflammation was paralleled by a reduction of inflammation at a systemic level. These are the results of pro-inflammatory cytokine in the plasma of the mice, and as we could see from this data, the high fatty sugar consumption evoked an increase of all the pro-inflammatory cytokine that TBB was able to counteract. In conclusion, we could see how it is possible to targeting meta-inflammation, identifying a kinase, casein kinase 2, which is directly involved in the activation of other well-known pro-inflammatory signaling cascades, and how the TBB administration was effective in ameliorating the diet-induced metabolic disorder. In conclusion, I want to thank Professor Massimo Collino from the University of Turin, Professor Carlo Cifani from the University of Camerino, my colleagues for the support, and thank you for the attention.
Thank you, Dr. Porchietto. And the paper is open for questions.
My name is Osrög, I'm Geneva University. It's very simple structure, and the affinity to the CB enzyme is not very high, I guess. So you have used fairly high doses. Is there no better, maybe selective inhibitors of casein kinase?
There are many other inhibitors of CK2. It depends on their action. I selected a CK2 inhibitor, is an ATP competitive inhibitor, according to ATP,
course,
yes.
Yeah.
And the dosage was selected according to previous in vivo study that using chronic model this molecule.
Yes, so it's probably blocks other kinases as well.
It's pretty selective, but it could be that we selected one of the most potent one that we could find.
Yeah. The first slide you showed on the increase of publications on kinase inhibitors and on metabolic syndrome. Yeah. There was a correlation. That was not this type of kinase. That was
no, just general kinase. Yeah.
Okay. Yeah. Good.
Yeah.
Thank you. It's very well structured and nice work.
Thank you.
Yeah.
Further questions? I have one. Have you checked the contribution of other lipids on this model, such as cholesterol or triglycerides, and the effects of your CK2 inhibitor?
Thank you for the question. We just evaluate the total cholesterol and triglyceride level at serum level, but we not evaluate the signaling pathway correlated to cholesterol synthesis or triglycerides.
It was lower?
Yeah.
Okay.
Yeah.
Any further questions? Okay, thank you very much for your presentation. And I'd like now to introduce next speaker. Sorry, I have to wait for Next speaker is Dr. Zhang-Yin from the Huazhong University of Science and Technology, who's presenting the paper Platelet GPVI is a mediator of rhabdomyolysis induced acute kidney injury in mice. Thank you, Doctor.
Thanks, Chair. Good afternoon, everyone. Today, my presentation is platelet GP6 is mediator of rhabdomyolysis-induced acute kidney injury in mice. I'm Zhang-Yin from Tongji Medical College, Wuhan, China. Acute kidney injury, AKI, its characteristics is high incidence, is high mortality, and high hospital costs, and poor recovery. Because of low effective treatment now, so AKI is getting more and more attention from world. Rhabdomyolysis-induced AKI, it is RM AKI, is induced by muscular damage, and the rhabdomyolysis release myoglobin, introduce intratumoral obstruction. Myoglobin is also in the NO scattering, lipid peroxidation, apoptosis, NF-kappa B, and TGF-beta release. All of this needs vasoconstriction, oxidative stressors, inflammation, and fibrosis. As you know, platelet is the smallest cell in the blood. It is resting form under normal condition. But when the vascular damaged, the platelet activated and involved in hemostasis and thrombosis. Beyond this, it's also involved in other disease pathology, such as inflammation, injury, cancer, and so on. In the inflammation, platelet interaction with neutral cells. Neutral cells, monocytes, as this interaction promote inflammation. And between platelet and leukocyte interaction have direct and indirect interactions. The direct interaction involves platelets receptor and the leukocyte receptor, the molecular pair, such as P-selectin on platelet and the PSGR1 in leukocyte. Besides this, other receptors also involved it. Besides this, after platelet activation, some soluble factors, such as soluble CD40 ligand and other chemokines, cytokines, and indirect make platelet-leukocyte interaction. And the GP6 is a platelet and macrocyte receptor, only expressed in platelet and macrocyte. It have extracellular domain, transmembrane domain, and cytotoxic domain. When the collagen, the GP6 ligand, binding GP6 and trigger the signal transduction, make the Fc receptor gamma chain item phosphorylation and signal transduction, make calcium increased and platelet activation and release. Have reported platelet GP6 is involved in many diseases, such as phenolmier-derived sepsis, myocardial ischemic peripheral injury, cutaneous inflammation, the glomerulus, and cirrhosis. So, our project aims is to check whether platelet GP6 is involved in RMAKI and explore how does hemi contribute to platelet activation and RMAKI, and check if blockade of GP6 elevate RMAKI. First, we histologically the RMAKI mice model and check the biological parameters, BUN and the creatine increased. This implied the model is done. And we find in the RMAKI model have some platelet in the initial and in the tubular cells. So this implies platelets participate in the RMACI. And then we checked the model mice platelet function and found in model mice the platelet function, collagen-induced and ADP thrombin also increased compared with control. And the granule release is also enhanced. Other functions is clot retraction is enhanced also. And in model mice platelet, the Jola-I binding and the predilection dispersion also increased. This implies there are some substances in the blood of model mice where active platelets. And the hemi is from hemolysis or tissue injury damage from hemoglobin or microglobin. It's hemi. Besides its effect in other cells, it also involved in some disease. For example, the ACI. So we check if hemi induced platelet accretion. We use in vitro experiment dose-dependent hemi induced platelet aggregation and release. In vitro experiment, hemi also increased Jola-I binding and the predilection expression in platelets, in washed platelets. So this implies hemi active platelets and directly induced platelet aggregation and release. But why? We found in RMACI mice, GP6 expression increased and enhanced the downstream signaling. We can say, compared with control, GP6 increased. This is the total protein. And also, but other receptors like alpha-2B and P2Y12, no difference. And the downstream signaling, 6-phosphorylation, is also increased. So the hemi may be not only increase level of platelet GP6, but also increase the downstream signaling. So we use different hemi, check the watch the platelet signaling from GP6 downstream. We found the PRC gamma 2 and SIC also increased by the hemi. And in the watch platelet function aggregation, we can find like hemi, like collagen, increased the aggregation. But we use GP6 inhibitor, JAK1, like collagen. It blocks hemi-induced aggregation. So this implies hemi activates platelet via GP6 and enhancing the downstream signaling. And in vitro experiment, hemi also increased the platelet GP6, but to alpha-2B, P2Y12, other receptors, no difference. So we can say hemi specifically increased platelet GP6. And we use experiment in vivo experiment, JAK1, day1, this is GP6 inhibitor, and day4, make model, and day5, harvest blood and kidneys, we find the chemical BUN and creatinine decreased after inhibitor GP6. And from the hematologic, we found after use JAK1, GP6 inhibitor, the tubular injury score also decreased. So platelet GP6 promotes injury in RMAKI mice, and blocked GP6 can elevate kidney damage. Macrophage X traps, like neutrophil, also extracellular traps. Macrophage, when damaged, and platelet activation, and used with AKI, the macrophage DNA and histone released and makes mats. So we check GP6, if GP6 contribute to the mass formation. Compare with the control, inhibit JK1, inhibit GP6 with JK1, and decreased mass formation. And we compare with resin platelets. Resin platelets activation, the mass increased. But when you use GP6 blocker, the mass decreased. So platelet GP6 contribute to the formation of mats. In the summary, rhabdomyolysis and induce hemi release, and hemi active platelet from GP6 and make macrophage activation and forming mats, make tubular injury and induce the AKI. When use GP6 antibody JAK1 block GP6 and inhibit mass formation and elevate AKI. So this also thanks the Division of Nephrology, Tongji Hospital, Professor Zhang-Yin. Thanks.
Thank you, Dr. Ming. And the paper is open for discussion.
Hi, very excellent job. I know your research field focus on platelet, and platelet can secrete TGF-beta, and it can promote cancer metastasis. So how do you do related work on that part? And platelet secrete TGF-beta also promote tumor metastasis.
Thank you very much.
Yes.
It's a good question. Yeah, platelet can release TGF-beta, and it is maybe 50% in the blood. So it is also promote cancer metastasis. Our lab will submission a paper about this.
Yeah. Yes.
Thank you.
Yes. Thank you.
Steve Watson, University of Birmingham. That was a very impressive presentation, a lot of data.
Thank you.
The demonstration for involvement of GP6 is very clear, and JAK1 is very specific to GP6, but heme has also been shown to activate platelets through the C-type lectin receptor CLEC2. How do you think that is contributing here?
You mean the CLEC2 is also involved in this process?
There are two papers showing that heme is a selective ligand for CLEC2 on platelets, so it is also activating.
Yeah, I think so. Besides GP6 and another receptor, CLEC2 also involved in the process. Have a paper have published. This paper is done 2019 before the passing COVID-19. So, but the COVID-19 has delayed the publication.
Okay. And then just a second question. You showed an increase on GP6 expression on platelets. What was the time course of that increase? How long did it take before the GP6 levels were increased?
Sorry?
I think you showed midway in the talk, if I followed you correctly, that the levels of GP6 on platelets in the model increased,
Yeah.
not only the function but the level. How long did it take for that increase in GP6? How many days upon the induction of injury?
In vitro experiment?
Yeah.
In vitro experiment, we incubate hemi with 6 hours, and 6 hours is increased. Maybe I'm not sure how long, but 6 hours is also increased.
Okay, I think we can leave it there. I'm not sure, since platelets aren't making new protein, it's not on intracellular sites. It looks as if it's somehow becoming more accessible, but I'm not sure how you can see such a change in that time. But very, very nice work. Thank you.
Okay, thank you.
Hi, I'm Steve McWilliam. I'm a senior lecturer from Liverpool.
Okay.
One of the other proposed mechanisms for the kidney damage in rhabdomyolysis is direct sort of damage of the heme or iron on proximal tubule cells, so it gets filtered and then it has a direct impact. Do you think that there are maybe multiple factors going on causing the kidney damage, or do you think that the mechanism you're proposing is the main one?
Yeah, but AKI, different model, different mechanisms. We just use the rhabdomyolysis induced AKI. This is maybe hemi is the main point.
Which brings me on to my other question. So we see AKI much more commonly, or rhabdomyolysis is quite rare in our patients. The most common cause of AKI is post-cardiac bypass surgery, and we're beginning to think that maybe there's more of a role of hemolysis rather than just ischemia reperfusion in those patients. Have you done any work looking at the role of hemolysis itself and AKI?
Yeah, maybe ischemia is more common. Ischemia reparation induce the AKI.
Yeah, so we're just wondering whether there's a similar mechanism between hemolysis in the blood and rhabdomyolysis. I don't know if you've got any thoughts on that.
In the ischemia and reparation AKI, platelets also involved in that, but it's different mechanisms. Maybe has a paper 2 years or 3 years ago published. Yeah.
Okay. Thank you.
Thank you.
Thank you.
Any other question? I would like just one more question to ask, which is, have you measured the concentration of heme that you have in your model of rhabdomyolysis, just to be sure that it's in the same range of the concentrations that you use in your experiments?
Oh, yeah. Different laboratories, different dose. Yeah, we reference a paper, nuclear medicine. It is this dose. Some laboratory is lower. I'm not sure which one. Maybe I think maybe is correlation the damage never. The damage never use. If it's heavy, maybe high dose, high concentration. Is it nurse maybe low concentration.
But you didn't measure in your rhabdomyolysis model. You haven't.
No.
Okay.
Yeah. Thank you.
Okay. Thank you very much. And we are closing this session. I would like to thank all the speakers as well as the audience for their participation. And that's it. Thank you.